Welcome to The Explainer. Today, we're diving into a premium, structured tutorial all about the incredible science of how we actually decode plant diets. Have you ever looked at a massive, thriving tomato plant and wondered exactly what invisible building blocks it pulled out of the earth to grow? We usually just take it for granted that plants need dirt, right? But science requires precision. So today, we're going to explore exactly how researchers literally stripped away the dirt to uncover the exact microscopic nutritional needs of the world's crops. So here's the core scientific mystery we're dealing with. How do we figure out what a plant needs to survive if the soil it sits in is already this messy, unreadable soup of minerals? To figure out if a specific element, let's say zinc or magnesium, is essential for a plant, scientists need a way to grow that plant with all the nutrients present except for the one being tested. But you just can't isolate and test a single nutrient in soil, because the dirt is already full of them. It's kind of like trying to figure out the secret ingredient in a complex stew that's already been cooked. You just can't do it.
Here's our roadmap for today's explainer. We'll start by moving beyond the complex soil, then cover the basics of hydroponics, dive into the different types of hydroponic systems, look at historic nutrient solutions, and finally, unpack modern media and chelators.
All right, Section 1, Beyond the Complex Soil, the problem with dirt. Let's jump in.
See, the ultimate obstacle for those 19th century botanists was that soil is essentially a messy soup. If you want to know exactly what a plant is eating, the soil is actually your enemy because it completely hides the data. To solve this, early scientists realized they had to eliminate soil entirely. They replaced it with the ultimate blank canvas, pure water. By using water, scientists could control absolutely every single element added to the plant's environment. If the plant died, well, they knew exactly what was missing. Historically, this whole process evolved a lot like building blocks. Back in the 19th century, pioneering botanists like Julius von Sachs and Wilhelm Knop completely changed the game. They invented what they called solution culture, which meant growing plants with their roots suspended in a solution of inorganic salts and water, with absolutely no soil whatsoever. This foundational work eventually led to 1938, when Dennis Hogland at UC Berkeley formulated a highly precise medium that supplied all known essential nutrients. And today, we've taken Hogland's breakthrough and engineered advanced, modified solutions that power everything from standard laboratory research to massive commercial indoor farms.
Which brings us to Section 2, the basics of hydroponics, growing without soil.
So the crucial scientific solution here is hydroponics. By definition, it's the method of growing plants in a nutrient-rich solution completely without soil. Those early pioneers proved a really profound foundational truth of biology. Plants don't actually need dirt. They just need the mineral elements that the dirt holds, along with water, air, and sunlight. If you can provide those things directly, the dirt basically becomes obsolete.
But listen, you can't just drop a plant in a glass of tap water and walk away. There are some absolute non-negotiables for this technique to actually work. First off, as the plant eats, it changes the chemistry of the water. So you either need a massive volume of solution, or you have to frequently adjust the pH and the mineral concentration. Second, and this is a really big one, plant roots need to breathe. They need oxygen. If they just sit in stagnant water, they'll literally suffocate and drown. That's why vigorous oxygenation, usually through porous air stones that bubble air into the water, is absolutely required. And when you get all these factors dialed in, the commercial viability is immense. This exact science is how massive greenhouses pump out flawless tomatoes, cucumbers, and cannabis all year round.
Moving right along to Section 3, Types of Hydroponic Systems, Engineering the Environment.
Mechanically speaking, there are three primary ways to engineer this soil-free root environment. You've got deep water culture, where the plant stems are suspended right above a tank, and the roots hang directly down into heavily aerated water. Then, you've got substrate-based systems, like a drip system. Here the roots are supported by something totally inert, think sand, gravel, or rock wool, and the nutrient solution is dripped right onto them. Finally, you have aeroponics, where the roots are just dangling in the empty air, and they're continuously misted with a nutrient spray.
Now, one of the absolute most popular and efficient designs is the nutrient film technique, or NFT. The cycle starts when a pump pulls the nutrient solution up from a main reservoir. Then, that solution flows in a very thin, continuous layer down a slightly tilted trough. The plants just sit in this trough, and their roots are bathed in this constant moving film of water. Because the layer is so incredibly thin, it provides plenty of oxygen alongside the nutrients. After that, the water returns to the reservoir to be pumped again. It's this beautiful continuous cycle. And that's pretty different from, say, ebb and flow systems, which periodically flood the roots and then completely drain the water away to let them breathe. It's really fascinating to see just how highly technical or wonderfully simple these setups can get. On one hand, you have active systems like aeroponics. This requires water pumps, specialized misting nozzles, timers, and higher nutrient concentrations. It's incredibly precise for sure, but technically demanding. On the other hand, you've got passive systems, specifically the wick system. This is literally as simple as it gets. You just use a wick, like a felt strip or a simple string, to draw nutrient water up from a reservoir right into the growing medium. It works entirely through capillary action. Zero moving parts and absolutely zero electricity required. Okay, now that we understand the hardware, let's jump into Section 4, Historic Nutrient Solutions. Time to talk about perfecting the actual recipe.
Think of this history like an evolving family recipe. Take Knopp's solution from the 19th century. Wilhelm Knopp used just 5 key mineral salts, and it worked. But honestly, it was a bit of an accidental success. His chemicals were highly impure. They were contaminated with trace micronutrients that he didn't even know existed. But as it turned out, the plants secretly needed them. Fast forward to the master chef, Dennis Hogland. By 1938, science had advanced, and Hogland formulated a precise medium that comprehensively split out all the known macronutrients, like nitrogen and potassium, from the vital micronutrients, like zinc, copper, and manganese. It was a massive leap forward.
Which brings us to our final section, Section 5, Modern Media and Chelators, Fixing the Chemistry.
Hogland's recipe was brilliant, no doubt, but modern science found a few chemical limitations that really needed fixing. First up, Hogland relied primarily on nitrate for his nitrogen, but as plants absorb nitrate, it can cause some wild, dangerous swings in the water's pH. Today, we balance the recipe by adding a bit of ammonium. Because the plant absorbs both positively charged ammonium and negatively charged nitrate, the overall pH stays nice and stable. We also use higher initial concentrations of nutrients, and we add beneficial elements like silicon, which actually helps reinforce cell walls and makes the plants way more resilient. But the single biggest upgrade, that was the introduction of synthetic chelators like EDTA.
Okay, we really need to dive into this because keeping iron available to plants is actually a huge chemical headache. See, iron easily precipitates out of water, meaning it turns solid and just sinks right to the bottom, especially if the water is alkaline. If that happens, the plant's roots can't absorb it and the plant essentially starves. Enter the chelator. A chelator is a molecule that binds to metal ions, forming a stable, soluble complex. Early researchers tried using simple citric acid, but today we rely on advanced synthetic chemicals like EDTA or DTPA. I love this analogy. Think of the chelator as a microscopic cage. It literally locks around the iron ion. This cage prevents the iron from reacting with other elements in the water and turning into a useless solid. The iron stays dissolved, it stays suspended, and most importantly, it stays perfectly available for the plant's roots to absorb. And here's the coolest part. Once the root absorbs the iron, the empty chelator cage actually diffuses back into the water to go hunt down and bind with another metal ion. It's this elegant microscopic delivery system that keeps the whole hydroponic process alive and thriving. So we've gone all the way from messy, unpredictable dirt to perfect, mathematically balanced liquid diets in highly engineered environments. I want to leave you with this final thought. If we can now perfectly engineer a plant's exact diet without using a single grain of soil, what does the future of global agriculture actually look like? As our climate changes and arable land becomes more and more scarce, could the blank canvas of hydroponics be the ultimate solution to feeding the entire world? It's a deeply fascinating question, and honestly one we are already beginning to answer. Thanks so much for joining me on this explainer, and keep learning.